Paravascular exchange of cerebrospinal fluid and interstitial fluid. Paravascular spaces are formed by the ensheathment of leptomeningeal vessels by the pia within the wider subarachnoid space. This creates a distinct compartment between the pial membrane and the wall of the blood vessel. The courses of paravascular spaces surrounding arteries and veins differ from one another with important consequences. The paravascular spaces surrounding arteries penetrating from the cortical surface follow the vessel as it enters the brain parenchyma. As the vessels branch at the arteriolar level, this paravascular sheath becomes discontinuous, and the paravascular space merges with the basement membrane of vascular mural cells. In contrast, the pial sheath that surrounds veins is reflected at the cortical surface and does not proceed into the brain parenchyma. Thus, the paravascular spaces surrounding veins at the cortical surface is continuous with the subpial space and the paravascular spaces surrounding interparenchymal veins. The movement of CSF from the cisternal compartments into and through the brain interstitium has been explored in the intact brain of mice using an imaging approach called in vivo two-photon microscopy. When green fluorescent tracer is infused into the CSF of the cisterna magna, this tracer moves along the cortical surface within spaces immediately surrounding cerebral surface arteries. In these images, the blood is labeled in red. It is clear that the CSF tracer is moving rapidly through the paravascular spaces surrounding these surface arteries. When CSF tracer is imaged below the cortical surface, it becomes apparent that the CSF moves into the brain along the paravascular spaces surrounding penetrating arteries, indicated here with arrows, as they dive into the brain parenchyma. From here, green CSF tracer moves readily into the surrounding interstitial spaces. The magnitude and rate of this paravascular influx of CSF into and through the brain interstitium is evident when in vivo two-photon microscopy is employed to conduct time-lapse imaging. At the cortical surface, CSF tracer moves rapidly along the paravascular spaces surrounding surface arteries, collecting around penetrating arteries, and eventually invading the subpial space. Below the cortical surface, tracer first enters the parenchyma along paravascular spaces surrounding penetrating arteries, then spreads outward into the surrounding interstitium. While in vivo two-photon microscopy is limited to a restricted but high-resolution view of the cerebral cortex, the exchange of cerebral spinal fluid and interstitial fluid along paravascular pathways can be imaged throughout the brain by dynamic contrast-enhanced MRI. In these studies, rats were imaged in a 9.4 Tesla MRI scanner. An MRI contrast agent was infused into the CSF of the cisterna magna. Dynamic imaging shows that the CSF-borne contrast agent moves rapidly through the cisternal network, then over the brain surface along paravascular spaces surrounding large arteries that appear contrast negative. This imaging shows that the rapid movement of CSF from the cisternal compartments along paravascular spaces is a brain-wide phenomenon and contributes to the extensive and rapid exchange of CSF and interstitial fluid throughout the brain parenchyma.